As Trinidad and Tobago enters a new phase of the pandemic, the vaccine stage, welcome to A Small Dose of Hope can bring us together here on CNC3, an ANSA McAL series that examines the benefits of the COVID-19 vaccine. My name is Francesca Hawkins. My guest today has been at the forefront of the region's effort to get on top of the COVID-19 pandemic. Professor Christine Carrington is Professor of Molecular Genetics and Virology at the Department of Preclinical Sciences at the University of the West Indies campus at St. Augustine here in Trinidad. Professor Carrington's extensive research work has focused on several viruses, all familiar to us here in Trinidad and Tobago, such as dengue, rabies, yellow fever, Zika, and the chikungunya viruses. Her published work on coronaviruses dates far back as 2008. She is now the principal investigator for the COVID-19 Impact Project. It's a collaboration and a knowledge-sharing hub between UWI, the Ministry of Health, CARPHA, University of Oxford, and the University of London. It's our great privilege to welcome Professor Carrington. Professor, as a scientist who's dedicated the last year of your work to stopping the spread of COVID-19, so to speak, I'd like to start by asking you, what does it mean to you personally, professionally as well, to see the start of the vaccine rollout here in Trinidad and Tobago? Professor.
Oh, thanks so much for having me on. Well, the first thing to say is that I watched the vaccine rollout yesterday. I saw the first vaccine going to the first Trinidad and Tobago arm, and it was really I found it very moving. The vaccine wasn't for me, but I felt a sense of relief and optimism. I am very excited about the rollout. I'm very excited about the potential for me and my family and my friends and everyone in Trinidad to get this opportunity to be protected against this virus. So it was really something I enjoyed.
It was moving for you. Now, going back approximately a year to January the 30th, the chief of the WHO announced to the world that the pandemic was here and that scientists around the world had a window of opportunity to work together to stop the pandemic. Now, at that time, you would have been working on other kinds of research. What did it signal to you and your team the minute you heard the WHO Director General make this global statement?
So at that time, much of the work I had been doing was with two of my graduate students. One of them was working on rabies, so I had my head in rabies literature, and the other one was working on chikungunya and Zika. So I was very involved in what she was doing. So when we first heard about this coronavirus, I mean, of course, I remembered that we had published some work on coronaviruses before. It's not a virus that I would have spent much time working on, but we had some knowledge. And when it first came, I immediately knew that we had to at least be able to set up systems to be able to detect that virus. And so, very early on, we ended up working with the Ministry of Health, supporting them in the rollout of the diagnostics, setting up their technology for diagnostics and all of that. So we as virologists, it was interesting. It was not unexpected because, we've been having more and more new diseases emerging into human populations from animal populations. And we had Zika, all the other ones before, chikungunya, and so it wasn't a surprise.
It was unprecedented in medical history, the speed at which scientists globally pivoted, so to speak, from their research towards the coronavirus research. Can you tell us the steps which the local team experienced in terms of the collaboration, the information sharing hub that was developed between yourselves at UE, CARPHA, as well as the Oxford University, and so on and so forth? Can you tell us what that pivot was like to go from a local focus to now collaborating intensively with global scientists from the University of the West Indies?
Right. So from the beginning, as an individual scientist and with my group, we have always been interested in and understood the power of sequencing of virus genomes to, being able to work out the structure of the genetic material of the virus and what sort of information you can get from elucidating that. It's more than just being able to characterize a virus. There's so much more you can learn about it, about the virus and the epidemic and everything. So we knew that that was all possible. We had been working very closely with people at, in the past with people at the University of Oxford and all these other places. So it wasn't until about probably around March or April that we started formulating this project, COVID-19 impact. And we actually didn't get, we applied for funding around May. There was a funding opportunity, and it took a while for us to get the money and everything and get things set up. But what has impressed me is the, scientists have always been collaborative, but there's also an element of competition as well. And I think with this whole COVID-19 pandemic, it's just pivoted straight into complete collaboration and openness. I could pick up, text somebody in the UK and say, look, I'm having a problem with this. What do you recommend? And they would say, oh, I can't help you there, but, try so and so. And they put me on to the next person. So it's been very, very collaborative. The ministry has been very helpful, as well as working closely with CARFA. It's just been a free flow of information and very, very encouraging and very collaborative.
So in some respects,
and I think a lot of scientists around the world have experienced that. It's not just us here. You don't feel like you're in an outpost. You feel integrated.
So in some respects, the COVID has brought out some aspects of the best of humanity, would you say?
I would think so. Certainly in the sciences. Just the simple act of sharing the when the virus was first sequenced, when they worked out the sequence for that viral genome, the simple act of making that available to all scientists was the beginning of this incredible, this incredible rush of science and development and technology, and this whole movement that has allowed us to have a vaccine, to have several vaccines in 11 months. We had of this vaccine. So it's that open data sharing is a very important component. When, for example, when we sequence viruses, we get samples from CARFA or the Ministry of Health, and we sequence them. What we're doing is we're looking to see if there are any variants of concern. I'm sure everybody has heard about them. And whether we find them or not, whatever sequence we generate, we add it to the global database of knowledge so that other scientists can see it as well, and our information can be incorporated into studies elsewhere. So it's all part of a big collaborative effort. It's been very, very encouraging.
When it was first announced in Trinidad and Tobago that the UK variant had arrived, there was a measure of anxiety across the society. And then this was generally changing because confidence was now in the society when we heard that right at UWI the variant had been identified. And this was very much part of your work in the sequencing. Can you explain to us what your capacity is in terms of spotting the mutations, and how does that surveillance help us here in Trinidad and Tobago to manage the pandemic and, of course, across the region?
So there's a variety of things you touched on there. So the first thing is you mentioned what did we think when, what people's panic when it first was announced that this UK variant, which is actually called B117, it's a much more cumbersome name, but we try as far as possible to avoid, labeling viruses with countries and stigmatizing those countries. So when that variant first arrived, the biggest relief was finding out that it was not in someone from the general public, that it was in an individual who had been quarantined. So that meant that, yes, we had detected it, but it wasn't out there spreading. And I think that was that for me was what was most important, that the systems that we have in place to ensure that we don't, to limit and mitigate against spread, to prevent introductions, that those were working. So this was not someone who was out there with the capacity to spread it to other people. So that was the first relief. In terms of the capacity that we have introduced, the technology we've introduced is called nanopore sequencing. It is a very rapid and very flexible technology. The actual sequencing device is a little small thing. It's about the size of a stapler, and it has a USB connection to your computer. And so it's very flexible, it's portable, and in each one of those runs that we do on that little device, it's called a Minion, Oxford Nanopore Minion device. Every run that we do, we can run 24 samples at a time. We have, so we can get that in about a day. We can have 24, the results from 24 samples. And we are hoping to increase that capacity. Now, when we do sequencing, we are not looking, we basically get the genetic material of an organism is a sort of sequence of, let's say, letters, 4 letters that change in sequence. And we're working out the full sequence for each that is found in the virus in each sample. When you have that full sequence, you can work out exactly what lineage, what that variant belongs to. Some of those lineages we are concerned about. So we're getting more information than just, yes, it's a variant of concern, or yes, it's the UK variant, or yes, no, it's not the South African variant. There's lots more we can look at there. We can also go back to the data that we have to look, if we get, we're going along, we're looking for these 3 major variants, the one that was first detected in the UK, the one that was first detected in South Africa, and the one that was first detected in Brazil. So those are the ones we're keeping an eye out for. But as we go along, other ones come up in the news. There's other mutations we have to look out for, and we can always go back and look at the data we have. So it's quite, it's a very robust and flexible system, a flexible approach to surveillance.
With as much knowledge as you're describing about the virus and its sequencing, can you touch then on how the vaccine is interplaying with all this new knowledge? It's of great interest here in Trinidad, especially as the vaccine has begun its rollout.
Right. So one of the major reasons that we have arrived so fast at so many vaccines is that we got the sequence for that virus that was first identified in Wuhan. So that sequence was released. We knew what the genetic material of the virus looked like. That allowed us to know what the virus's proteins look like and which proteins were most likely to be useful components of a vaccine. So that's the first thing it gave us, the sequencing. Now, obviously, what we've seen now is that when you start off, viruses, they evolve over time. Like anything else, they evolve. And so at the beginning of this pandemic, the people who were infected, or more or less the vaccine in them looked pretty much the same from one person to the next. But over time, the virus sort of diverges. You get new lineages emerging, like branches in a family tree. And some of those lineages seem to be less easily neutralized, or the efficacy of the vaccines is lower against them. So we know that. However, it's important to note that the trials that were done and what they've shown is that the vaccines may be less efficacious against preventing mild and moderate disease, but we don't know yet. And there's some suggestion that they still protect against severe disease and death. So we still have to find that out. So it's not a complete loss. You do get a reduction in the efficacy, but it's not a complete loss. There's still some protection, and we hope protection against at least the most severe manifestations. So we can protect against hospitalization, and we can protect against deaths. So it's not all lost, but we want to get out there fast, with our vaccination campaign.
You're watching A Small Dose of Hope Can Bring Us Together. It's a series that examines the benefits of the new COVID-19 vaccine. Our guest is Professor Christine Carrington, the principal investigator for the COVID-19 Impact Project. It's a collaboration and knowledge-sharing hub between UWI, the Ministry of Health, CARPHA, the University of Oxford, and the University of London. Professor Carrington, you were just explaining to us about the new data that's coming in about the efficacy of the vaccine. Can you tell us still why it's still important to take the vaccine, although some of the data is unfolding? Why is it still so important that we adopt the rollout and volunteer for taking the vaccine?
Okay, so these vaccines are our way out of this pandemic. The purpose of a vaccine is to prevent a person from getting ill or prevent illness or death without getting the actual disease that you would normally get, that the vaccine is protecting against. So normally when we get infected, our body produces an immune response, and that immune response can protect us against future infection with that same virus. So what we've done now with a vaccine is the vaccine basically tricks your body into believing that it's met the virus, it mounts an immune response, and then going forward, should you ever encounter that virus again, it can mount a new immune response very, very quickly and protect you against the worst effects of this disease. Now, ideally, you would love a vaccine that prevents infection altogether, okay? But most vaccines don't prevent infection totally, but what they do prevent is disease. And when people talk about the efficacy of these vaccines, when you hear that the efficacy of the AstraZeneca vaccine is about 70%, or the Pfizer is 94% and stuff like that, when you hear that, what they're saying is the extent to which they protect you against disease. Okay, if we look, so there's a vast difference. If you think of your chances against the virus, the chance of getting, you have a 90% less chance or 70% less chance of getting disease if you take this vaccine. So that's a big difference. Also, even if you are one of the people who takes the vaccine but still manages to get sick, what the data has shown is that the severity of your disease is going to be much, 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 much less. These vaccines, they're protected totally, some of them protected totally against severe disease.
Can you explain to us the ways in which the vaccine will protect you against severe cases of disease? How does that work? How can it protect you against serious cases of disease, but less so for mild cases? Can you explain that?
So when you get infected with a virus, the virus gets into your body, it gets into your cells, and it starts to multiply, and the levels of virus go up. And as the levels of virus go up, in some diseases, what you get is damage being done to your cells and tissues and stuff, and that is what causes the symptoms. Now, your immune system, as the virus goes up, your immune system kicks in, and that brings the virus levels back down, right? And then that's, so the damage, your body has time to heal and get over that damage. Now, in some viruses, for example, the SARS-CoV-2 virus, a lot of the damage can be caused by not just the virus itself, but by the subsequent immune response, not to the vaccine, but to the other parts of the immune response can also cause damage along with the virus staying there. So the faster your immune response can dampen down on that virus and prevent it from damaging cells, prevent it from damaging tissues, prevent it from spreading, the less symptoms you will likely have. Okay? Now, severity is not just about the virus and the immune system. It's also about underlying disease. It's also about age. So some people are much more susceptible to severe disease than others. And if we can prevent high levels of virus in the person's body, prevent that extreme immune responses and things like that, we can protect against disease. Now, I should add that I am not a pathologist, so I'm not a medical doctor. So, by protecting yourself against infection, protecting against, dampening down that amount of virus, allowing for a rapid immune response, you protect yourself against more severe manifestations.
And what about the virus's strength in terms of protecting for reinfection or in fact spreading the virus?
Right. So as I said before, not all vaccines protect against infection. They protect against disease. And in this case, we're still learning about the extent to which the COVID-19 vaccines protect against infection and transmission. So it's possible you get vaccinated, you may be exposed to someone who's infected, The virus is introduced into your body, it may replicate, but not to the levels where you're going to get very sick. What we don't know is whether those levels that it replicates to and how it spreads in your body, if that then allows it to spread from one person to another. So that is why they've said we have to continue wearing masks, we have to continue social distancing. But I would add that the things are looking encouraging because there are at least two of the vaccines, including the AstraZeneca one, that the preliminary data suggests reduce the amount of infection and reduce transmission. So it's looking hopeful. And I think that, that's an important element. We don't just have to protect individuals. We want to protect populations. So being able to stop transmission is important. It's not essential, but it's important. You mentioned reinfection. That is an interesting thing as well. So over time, what we don't know for the vaccine yet, because of course we haven't gone on long enough, but what we do know from other coronaviruses that over time, our immunity for the viruses, it wanes. And there is the opportunity for reinfection. And a year into this thing, we're beginning to see reinfections with SARS-CoV-2. So there are people who were infected before and they had an immune response, but the immune response has waned and they have been reinfected. But what the data is showing so far is that those reinfections are largely mild. So you're not getting the, very severe, disease upon reinfection.
From a scientific point of view, you've been very open to share with us information about where the vaccine's efficacy is, as well as the great benefits. You said it was very moving for you to have seen the vaccine begin its rollout here in Trinidad and Tobago. So just as we're coming to the end of our conversation, can you go back there and reiterate once again the tremendous pluses for Trinidad and Tobago? Of course, your work is at the heart of it.
Okay, so the first thing I'll say is that these vaccines and the one that we have first gotten, these vaccines have been given to tens of millions of people all over the world, and they have been shown to be extremely safe and very efficacious. So there's no, I don't think anybody should have any hesitation about the safety of the vaccine or whether it's going to work, etc. I can't emphasize the importance of being protected against this disease. We have been locked down for months. We have children out of school. We have people not being able to work optimally. We've had to do these things. We've been kept away from our families. We have to do, and we have to continue for a while longer, but the vaccine is going to help to bring us back closer to a normal life. And we just have to go that little longer. We will get it done. And I just can't encourage people more. Their chances against the virus are much worse. And the vaccine is the alternative.
Professor Carrington, thank you so much for sharing your expert knowledge with us today. This has been A Small Dose of Hope Can Bring Us Together, a new series here on CNC3 presented by parent company ANSA McAL. Our guest has been Professor Christine Carrington. She is the principal investigator of the COVID-19 Impact Project, bringing together scientists from across the Caribbean and international counterparts. Our focus will continue to examine the benefits of the vaccines, specifically the COVID-19 vaccine, as we speak to renowned doctors, scientists, and medical thought leaders from Trinidad and Tobago, the region, and internationally. Thank you for joining us. See you next time.